do the thing where you like walk up to it?
I
like,
don't. It just automatically. It's awesome. All right, so we're going to talk about protein synthesis day 2, which is mRNA editing. So remember last time when we were talking about transcription, we built that mRNA strand, that little recipe card of our, like from our cookbook, from the original DNA. We copied down that little segment, and then we kind of cut it off, and it kind of floated away for a second in the nucleus while we finished up disassociating all of the proteins. Does that ring a bell?
Yeah.
Perfect. So now we're going to work on making sure that that little mRNA strand is ready to go out into the cytoplasm. So I'm going to write up here a sample of an mRNA strand with the actual nucleotides. And then we're going to use that to kind of build onto it and get it, at least starting to get it ready to leave the nucleus here. Let's change this one first. So in this case, I'm going to be using pink to represent our mRNA strand. I'm going to be adding other colors in order to say what we're going to add onto this strand. So pink is the original mRNA. Now, I'll give you guys another analogy. This little mRNA strand is going to be a little stick figure guy. Now, the mRNA strand is all nice and safe in the nucleus, but we got to get it out into the cytoplasm for us to actually do translation, which is where we're going to be building the proteins. So for this little stick figure guy here, that means that eventually we need to throw him out of the nice safe place, and he's going to end up kind of going into crocodile-infested waters, which is our cytoplasm. In the cytoplasm, there's a lot of enzymes that would love to break down this. We need to protect this little guy here first before we send him out into the cytoplasm. And we're going to protect him in 2 different ways. The first way is that we're going to give this little guy a nice helmet. Protect his head. Okay. The next thing that we're going to protect him with is a pair of little boots. So the idea with this is that, let's say that the crocodiles can't attack from the middle, they have to attack on either end. I know that's not how crocodiles work, but go with me here. So we can't attack from the middle, they have to attack from either the head or from the feet. In this case, the enzymes, it's really hard for them to attack in the middle. It's a lot easier than for them to attack either from the 5' end or from the 3' end. So we want to protect those 2 ends. Okay? So let's start with the helmet. To give this little mRNA strand a helmet, we are going to add on what's called a 5' cap. Cap like a helmet. This cap is actually going to be a guanine. But there's something funky about this guanine. So remember our nucleotides here. Terrible drawing. There we go. Remember, our nucleotides are facing in the 5' direction, so we have the phosphate kind of sticking out on this side here. And they're building, and all the nucleotides are facing in this way. It's single-stranded because it's an RNA. With this 5' cap, we're going to add on the guanine, but we're going to add it on backwards. add on the guanine facing backwards. And we're actually going to add another phosphate in between, so we have 3 phosphates bonded together. So if you think of like our ATP, our energy, it's very common to have 3 phosphates all kind of stuck together. We're going to try and do that here. It makes it stronger here so that it's harder to break off the guanine and attack the mRNA from the 5' side. So the first thing that we're going to do to protect this guy is add on a guanine. And this is actually called a methylated guanine, which is just a big fancy name that says this guanine has another carbon attached to it. So sometimes you see it being called a 5' cap or a guanine cap. Sometimes you see it being called a methylated guanine, just depending on where you're finding the information. Did I lose anybody? Alright. So now we got the helmet on this little guy. Let's give him some boots. On the other end here, on the 3' side, we're going to be adding what's called a poly-A tail. So think about it, poly-A tail is 3 words, so it goes on the 3' side. The poly-A tail is actually pretty easy. We're just going to slap on a bunch of adenines.
How many?
A lot. It could be upwards of 15 to 30 adenines that we're going to add on there. So it's just like somebody hit the A button and just like held down the A button on the keyboard and you get like that whole big stream of A's. That's kind of what we're going to be doing here. We're going to put on a poly-A tail. Now the thought with this is that if an alligator comes or a crocodile comes and he starts gnawing on the boots, he might take off a little chunk of boot, but that's okay because the foot is still there. He's not attacking the foot, he's not taking off a toe, he might just get a piece of rubber from the shoe. So if an enzyme comes and attacks from the 3' side, he might take off 1 or 2 of these adenines, but that's okay. We still have a bunch of other adenines, and more importantly, our pink mRNA strand is still safe and secure. It hasn't gotten deteriorated, it hasn't gotten broken down by this enzyme. Okay? Questions on this? You guys did awesome. That's the first part of my lecture on mRNA editing. There's a couple other things that we'll talk about on Friday as to how we're going to further prepare this in order to get sent out into the cytoplasm. But this is the first step in order to protect this little guy being out in the cytoplasm. Now the second part of the lecture today is on a codon. What a codon is it's basically a way to say the mRNA and organize it so that way we know how to build the protein later. A codon is a 3 nucleotide chunk of our mRNA. Now we're not adding anything here. We're just saying that these first 3 letters, we're going to kind of group them together into one codon. So it's a way to read the mRNA. And what the codon actually does is it's going to code for which amino acid we're going to add on when we're building our protein. Remember, proteins are a big, long string of amino acids, and each amino acid is like an ingredient that we need to put in the right order in order to make the protein that it not only works, but is in the same shape, and it's the protein that we wanted to make.
Wait, so what does the codon actually do?
The codon is just going to kind of identify these first 3, and it's going to code for a specific amino acid that we're going to put on later. the way that we know which amino acid is what we use called a codon wheel or a codon chart. Now we have a whole bunch of these in one of the cubbies over there. I think it's the second cubby on the right-hand side. But you guys can have one of these copies here and use this. We're going to keep working with this more on Friday and Tuesday as we continue on with our protein synthesis. But we're just going to kind of introduce it to you guys today so that you guys know how it works. So when we're looking at this, we'll look at the first codon first. And you always start in the middle. So start with A, then you go to U, and C actually says that it codes for an amino acid called isoleucine.
Wait, when you say start in the middle, you mean don't start with the caps, right? Like don't start with the methylated guanine and don't start with the poly.
Correct. We're just going to look at our pink mRNA strand.
Wait, just if it's a methylated guanine, it has to be guanine on that side.
Yep, it's always going to be a guanine.
And then on the other side, it's always going to be an adenine.
It's always going to be an adenine. Yep. So we always have the same cap and the same tail. The codons in the middle are what change, and that's what's actually going to tell us which amino acids to put in which order. So this first one here, we said that it coded for isoleucine. So again, I took the one from the middle first, which is A. The second letter is U, and the third letter is C, and that tells me exactly which amino acid it codes for. For the next one, GGA, we can start with the G in the middle. The second letter is also a G, and the third letter is A, which is glycine. And we can keep going so on and so forth to complete our mRNA and say which amino acids we're going to be adding.
Do we have to measure that, or I mean, not measure, memorize that for the finals?
You will have one of these to use to work with the final. I don't expect you to memorize the wheel. It's good to know a couple of them just so that you can quickly identify them. But you will have one of these wheels or the chart, I think, that you can use either side. I like using the wheel. I think it makes a little bit more sense for me. On the back side here is a chart, and this one you go on the first side, you say which the first base is. On the top here, you say which the second base is, and on the right-hand side, you say what the third base is. And you kind of find the meeting point of all three of those, and it tells you the same amino acid. I like using this one, but for some people, this makes a lot more sense. Okay? So as we're going through here, we'll continue working with this during mRNA splicing and translation. But the important thing is to kind of get used to this idea of what a codon is, and that there are three letters that are going to code for an amino acid. Now this can all change when we have mutations. And we have three specific mutations that are more frequent and that could happen. The first mutation is called an insertion. An insertion mutation is just when we're going to randomly add in another base. For some reason it doesn't transcribe right. We had an extra C thrown in there, and it added in where it's not supposed to be. The problem with this is that now it's changing our codons. So this one's still fine, and this one's still fine. But now our codon, instead of saying CCG, is saying CCC, which then changes all of the other codons afterwards. So we have a codon that goes here, and we have one that's kind of left over that it may or may not actually try and code that. Yes?
Are there going to be any that don't have an equal, like an even 3, won't be divisible by 3 evenly?
So if we have one left over, there's two things that could happen. Either when they're making the proteins, it could say, well, that's random, we don't need that, and it won't code for it. Or it could try and take 2 of these poly A tail ones and try and say, oh, well, they must have meant that that's an amino acid.
Yeah.
So it could mean something that either that one just doesn't get coded, or it could try and code something else. Remember, when we're changing the codons, we change the amino acids that it's saying that we need to include. So options with this is that we could either put in a totally random amino acid that we didn't mean to have in there, which could either make the protein a totally different protein or just make it non-functional. It's not the right shape. It doesn't do what we need to. So those are the two big ones, that it could either say that the protein's just not going to be made, or the protein that we are making is wrong. So that's one form of mutation.
Is that for only insertion, but like, is there ever going to be like a perfectly normal mRNA strand that is like 10 letters? Or no, that never.
Like 10 of these letters?
Yeah.
So with that, most of the time our mRNA, we want to keep it in 3-letter chunks so that we can code for amino acids. So when we're transcribing it, we want to transcribe the entire gene so that we can code for all of the amino acids that we need. We have a couple extras on there. They may or may not get coded. If it's an important amino acid that we need to have in order to make that protein functional, we want to make sure that those are coded. So we want to make sure that there's a 3-letter chunk there that will code for the correct amino acid. Yeah. Okay. So that's one form of mutation. The second form of mutation is called deletion. In deletion, all we're doing is, just like the name suggests, we're going to delete an mRNA nucleotide. For some reason, that one didn't get coded correctly. It didn't bond correctly. So we lost that nucleotide. Now again, it's going to change all of our codons. That one's still fine. So it accidentally deletes that one. We didn't add it in there. This codon then changes, and all of the codons afterwards. So again, we could have a codon that's going to code for a different amino acid, or it could tell the whole translation process to just stop, which would mean that our protein didn't get made. Now, there's one more mutation that's a pretty big one here, and that's called a substitution. A substitution, instead of what, this one was supposed to be a G that I just deleted, instead of having a G there, we accidentally put a C in. So now when we're looking at our codon chart, the codons are still the same here. We're still in our 3-letter chunks. That didn't shift any. This one is still coding for isoleucine. But this next one, when we're looking at the codon wheel, we go G, and then C, and then A. It's now coding for alanine. It's coding for an entirely different amino acid than what we want.
Is it always going to be like a G and a C change, or can it be an A and a U?
It could be an A and a U change. It could be a really random one, like this A changes to a G. So with substitution is just when we're adding in a different nucleotide than what we intended, what we need. Now sometimes these changes don't change which amino acid it's coding for, which is then fine. We still had a little mutation, but it didn't change the protein that we were making. So that's okay.
Wait, which one's the insertion and deletion?
A substitution,
Oh.
sometimes it changes this code, but what it's coding for might be the exact same amino acid, which is then okay. So you're saying that if I changed this one here and it's still coded for glycine? Yeah, that can absolutely happen. A lot of times that happens when we change this third one in the codon. When you take a look at the codon wheel, a lot of times the third one, all of those last letters all code for the same amino acid. So sometimes it's just the first two that are really important. Sometimes all three are needed to be perfect in order to get that one amino acid. So that's an option too. Do you guys have questions about either editing the mRNA and putting on the cap and the tail, or what a codon is and why we're using it?
We need the amino acids to like make our proteins, right?
Yes. Amino acid put in long chains and then folded and twisted in the right way makes a protein that we can use.
And that's why it's important for us to have the correct mRNA strands and not have any of these mutations.
That's why it's important. If any of these mutations do happen, it's not the end of the world. If it makes a protein that doesn't work, that's okay. We can go back to the DNA and make another copy of that same recipe card again and try it all over again.
Oh, so it's like if you make like a bad batch of cookies?
Yeah. If you make a bad batch of cookies, it's not the end of the world. You go back, you get the ingredients again, you start making them again. Hopefully they turn out better this time. Okay. Questions, comments, concerns? Wonderful. I will be adding this drawing up onto the board for the mRNA editing here today. I don't know if I'll get it done before class, but otherwise when you guys come in either before or after school, when you guys come in for class on Friday, that will be up there that you guys can use that and take pictures of that in order to study for your exam. Okay. Perfect.